So this week's lab, we're going to talk about genetics. The last couple weeks I have not done a recording like this, and I'm not sure if this is helpful or not for you, but I'm gonna do it anyway, hoping that it's helpful. So you should be going over genetics sometime in your lecture, whether you are already or soon will be. So this will be kind of a refresher to let you know some of the terminology before diving into the assignment and into your quiz. So genetics and inheritance. Inheritance is the transmission of genetic information from one generation to the next. So basically passing on the DNA from parents to child, and so we've talked about meiosis, mitosis and meiosis, and in meiosis the sexual reproduction. So these gametes are being made, and so the adult is basically kind of splitting their DNA in half, and each gamete has half of that DNA, which will then go on to fertilize the zygote with another individual's gamete to create a new individual. And this DNA that's being passed to the new generation is basically this inheritance. This genetic information that is being stored in the DNA are genes. And we're going to learn about how these genes are expressed from the different traits and differences that they might be on the different chromosomes. So, homologous chromosomes. One comes from your mother, one comes from your father. They are going to contain the same genes on them. So we're not looking at a karyotype right here, but basically this is one set of homologous chromosomes. In a human being, there are 23 pairs of homologous chromosomes. So this one could be, let's say randomly, chromosome 7. And so one of the chromosome 7 pair, one from the pair is from the mother, let's say the red one. One of them is from the father, let's say the blue one. And so, well, I guess they're actually labeled down there, maternal and paternal. And so this is what we mean by a homologous chromosome pair. And the genes that are found on these chromosomes are going to be for the same traits. So if your hair color is coming from genes that are found on chromosome 7, well then they would be found on both the red and the blue of the homologous chromosomes. The genes will be at the same location on these homologous chromosomes. An allele is a different form of the same gene. So that hair gene, the hair color gene that we were talking about, could be blonde, red, brown, black. Like it could be all these different types, all these different versions, all these different alleles. Now, I say this hair color is coming from more than one gene. It is not just one gene that is doing this. It is several different genes. But for purposes of this, the allele could be blonde or brown. And so those are two different alleles that particular gene could be. For every gene, you have a maternal and a paternal allele. These alleles could be the same. They both could be blonde. They both could be brown. Or they may be different. Blonde or brown. And then if we're talking about different genes, you could have different variations. So here we're looking at the homologous chromosomes where there are certain alleles, or I'm sorry, there are certain genes, gene C and gene D. And you can see that for gene C, well actually for both of them, but for gene C, the maternal chromosome, which is found over here on the left, it has a big C. When it comes to your paternal, the one from your father, it has a little c. When it comes to gene D, the maternal has a big D as the allele. That is the maternal. The paternal chromosome of the homologous pair has a small d as its allele. Now these genes could be, anything. They could be something that shows an outward appearance. When that trait could be skin color, hair color, eye color, or it could be something internal. It could be, but basically, don't forget what a gene is. The gene will transcribe and translate to a specific protein. And so that protein will be expressed. And then that protein goes and does what that protein does. And if this protein is going to change the color of your hair, well, then that is where that protein will be going, changing the color of your hair. And it could be, it doesn't have to be a very simple one-to-one. It could, go down the line in order to change the color of your hair. It might not be super simple. There's some terminology that we need to learn when it comes to the allele combinations. And so this is basically, the combination of the allele found on the mother, the chromosome from the mother, the chromosome from the father, whether they are the same or different. There are different terms here. If they are the same allele, whether that's big T or little t, they are going to be homozygous. Homozygous, homo meaning same. So they will be the same alleles. If they are different, which is found in the middle here, big T, little t, that is heterozygous because they are hetero meaning different. When an allele is always expressed. So this is kind of jumping into this next idea. Sometimes a particular allele will be dominant over the other. And so dominant means that when they show up together, the dominant is going to be the one that is expressed. It will mask the recessive. So usually dominant is going to be expressed by the capitalized letter, big T. Recessive is going to be small t. Now, this masking and expressing can be for different reasons. Lots of times, the recessive allele is a mutated allele that is basically non-functional. So if a flower, so big T, little t. Let's say big T is going to make the flower petals purple. Little t is going to make them white. And so when big T is present, the gene that is going to be transcribing, translating to create that protein will be a protein that makes the petals purple. But if little t is being expressed, little t is going to express, it's going to transcribe and translate a protein that is just non-functional. Non-functional protein that's being put into the petals will not create any different color, and so the white just happens to be the color that is present without that protein. Now, it doesn't always have to be that way. Sometimes the recessive gene is able to have a functioning protein, but a lot of the times it's not. So I digress. Dominant. When you have homozygous dominant, that means that homozygous, they're the same, dominant. They are both dominant. And so the allele, obviously that will be expressed, will be the dominant, the big T. Homozygous recessive, they are both small t's. And which one will be expressed? The small t will be expressed. Whatever, in this case, white. This whole purple being the big T, white being the small t, the white will be expressed. Heterozygous. So this is when they are different, big T, little t. And so which one will be expressed? Because the big T is dominant over the small t and they're both being expressed, the big T will be expressed. So let me rephrase that because I said the word expressed too many times. The big T will be expressed, the little t will be masked. because they are both present. A couple more terminologies here: genotype versus phenotype. Genotype, genome. So basically, genotype is the individual's DNA sequence, the genes themselves, the alleles will be the genotype. So for that last slide that we were looking at, big T, big T, the genotype is big T, big T. Now genotype could be, you could be looking at a lot of different string of alleles, and so it could be more complex than that. But a lot of the questions that we're going to ask, what is the genotype? It's basically, big T, big T, little t, little t. These are the genotypes. And the genotype is what is actual there, like the genes, the alleles, the DNA that's there. Phenotype is the visible result of the underlying genotype. So purple versus white. So if the genotype is big T, then the phenotype would be purple. The phenotype is the visible appearance of that individual. And so little t is the genotype. White would be the phenotype. When we observe patterns in the phenotype, and this is usually, this is what we can see for the most part. We can see your phenotype, but we don't know what your genotype is. Your phenotype can give clues to what your genotype is, but it's not 100%. Because, a homozygous recessive, so if your phenotype was white, well then I would pretty much know the only way that it can be white is little t, and so that's the homozygous recessive. So that way I do know what your genotype is by your phenotype, if I knew the whole which one was recessive and dominant. But if you're purple, purple petals on your flower, I don't know if you're homozygous dominant or if you're heterozygous, because both of those genotypes are going to be purple phenotype. So again, homozygous dominant, homozygous recessive, heterozygous, those are terms that can be used for genotype because we're talking about the actual alleles that are present. Continue on with some more examples of genotype and phenotype. Big F versus small f. So big F will equal lots of freckles. Little f will equal no freckles or very few. And so the genotype of big F, which is homozygous dominant, that genotype will have the phenotype of lots of freckles because big F is being expressed. In a heterozygous individual, the genotype is big F, small f. The phenotype is still lots of freckles because in a heterozygous situation, the big F is being expressed. This one might be a little confusing, but basically, I'm not really sure where they're going with this here. So if because we've already talked about heterozygous, homozygous dominant, or heterozygous. I guess it's basically saying whatever this is, this knot right here is going to be lots of freckles. But let's, we've already talked about it. There's no reason to have this row here. But homozygous recessive at the bottom, the genotype is little f, and the phenotype will be few to no freckles. Now, the father of genetics is contributed to Gregor Mendel. He did not know what DNA was. I mean, I'm not sure if he knew that the actual molecule existed, but even if he did know that, he had no idea that DNA, the sequencing of DNA that we've learned for the last couple labs, had to do with creating genes and uh, and creating proteins and things like that. But he did know, he learned looking at physical traits, the phenotypes of these organisms, and crossbreeding them with each other. He learned that there had to be some sort of two allele system. Mathematically, it just worked out that way. And so Gregor Mendel was an Austrian monk, not Australian. He was Austrian, and he was a scientist. He knew his math, he knew his physics, he knew his botany, and so he used these things together and studied these physical traits. He looked at the pea plants to kind of do his different studies to determine how parents transmit the genes to the offspring. And so again, he didn't know that genes were a sequence of DNA. Even if he used this term, it was more along the lines of a particular trait. Mendel's idea: every generation a plant inherits 2 units of information about a trait, one from each parent. And so this was determined mathematically just by doing the different crossbreedings that we're going to talk about. So these performed crossbreedings. When we talk about the breed of a particular plant or a particular animal, these are man-made. And so you can think about the breeds of a dog and how there are particular traits that go along with that particular breed of dog. And so over years, that breed of dog has become a purebred because those traits are always going to be there. And humans created these breeds of dogs. All of these dogs came from a wolf. And so through the thousands of years, humans have been able to, this wolf is a little smaller, and then that wolf over there is a little smaller. If I breed them together, the chances of getting a small offspring is going to be high, and so you keep doing that. If that's what you want, you just keep doing that over and over, or whatever these traits that you're looking for: a soft coat, a loud bark, big teeth, whatever these traits are that you're looking for. These are ways of doing man-made artificial selection. And when you learn about evolution, you'll learn about artificial selection. And so these particular breeds are going to have pure that trait. So if this particular breed is a purple pea plant, then that means, let's say big P, little p, the big P is going to be purple, the little p is going to be white. Well, if you have a breed of purple plants, no matter what, they're always going to be purple, then that means you've basically have bred out all the small peas. There are no small peas, whether you're getting your gene from your female mother or male father. They're only going to be big P. And so this breed will be purple, big P's all across the board. The other breed will be white, small P's all across the board. And so that is what we mean by when we talk about being a purebred. A breed is that they're going to be homozygous, either dominant or homozygous recessive. And so the homozygosity is going to be what is giving these particular breeds the 100% that the offspring will be producing whatever trait you're looking for. Now this is artificial, but when it comes to Mendel looking at these studies, it was a perfect situation because he would not have been able to do these studies as well as he did if he didn't have these purebreds to start with. So, this is how he didn't know in the beginning that something was pure dominant, pure recessive. It just worked out that way, and it was, very great for him. So he mated a homozygous dominant pea plant with a homozygous recessive pea plant. This is called the P generation. P generation stands for the parent generation. And the offspring that comes from that will be the F1 generation. Later we'll talk about the F2. So practicing genetic problem solving, we're going to use a Punnett square. And this is a way of creating a prediction of what the offspring will be when two individuals are crossed. And basically this is giving you the probability of what the offspring will be. So in the example below, we have big A and small a. Big A is creating red apples, small a gives green apples. So the homozygous dominant and a homozygous recessive will be mated. Now the apples won't be mated, it will be the trees that are producing these apples. So that means we're going to be crossing a big A, that's the homozygous dominant, small a, that's the homozygous recessive. We're going to be crossing these two individuals, mating them. And so in this Punnett square, you're going to write the alleles of one. Usually it's the female. We'll write that at the top. So big A. And then the other alleles from the gametes, from the other individual, will be small a. Now, we're going to be filling in this Punnett square. So you'll bring down, for this cell, you'll bring down the A, the big A, you'll bring over the small a. When it comes to this cell, you'll bring down the big A, you'll bring over the small a. So we did the first one, the second one, the third one, and the fourth one. And so usually, always I should say, we write it with the big A first. It's just the way it's done. And so 100% of the offspring that will be produced by these two are going to be big A, small a. That means 100% of them are going to be heterozygous. Both of those are genotypes, big A, small a, heterozygous. But the phenotype is going to be 100% red apples. So this is an F1 generation creating 100% 100% red apples, 100% big A, small a. Now, that doesn't really tell you much, does it? Because you didn't learn about which one was dominant, which one was recessive. So if you were Mendel and you started from scratch, every time I cross this purebred red with a purebred green, I'm getting all red offspring. Okay. Well, now let's cross two of the F1 generation. So that F1 generation that we saw before is heterozygous, big A, small a, big A, small a. So we write those two alleles of the one, the alleles of the other, and then we do the same thing. We fill in these cells. But now we're going to have a little bit of variety, a little bit of differences happening here. So with this cell, big A, big A. With this cell, big A, little a. This cell, big A from the top, little a over, and this cell is little a, little a. So we have some differences going on here. I am not sure if I click, I don't want it to go away. Yes. All right. So This is now the F2 generation because F2, we've crossed two of the F1s. So this next generation here is the F2 generation. And this is the generation that we're actually going to see the differences. We're going to see the recessive allele being expressed in the phenotype. And so then when we do some mathematics, we can figure out these ratios, and this is what Mendel was doing. So the phenotype, red apples to green apples, is 3-to-1. How do we know this? Well, big A is going to be red. Little a is green. So we've got one that's green. I'm sorry, one that's red. The heterozygous is also red. The heterozygous is also red. So that right here, 75% of the offspring will be red, whereas 25% of the offspring will be green. So I said 75%, 25%. I'm cool with that way of expressing, or you could say a ratio of 3-to-1. 3-to-1. And that is this phenotypic ratio. And the genotypic ratio, which I guess I'm not sure if that one's going to be written here or not, but basically it is a 1-to-2-to-1 because this big A is one genotype. Big A, small a, there's two of them, and then small a is one of them. So the phenotypic ratio is 3-to-1. The genotypic ratio is 1-to-2-to-1. And this is a monohybrid. So we are looking at mono, one allele. Whereas you can go, and I think our next slide is going to be looking at a dihybrid. You could go tri, quad, and they get way bigger. These Punnett squares get huge. So dihybrid cross, you're going to be following two traits. So we're going to be looking at seed shape and seed color. Seed shape, big R is round, the dominant. Little r, the recessive, is oval shaped. Seed color, big Y is yellow, and little y is going to be white. And so your P generation, just like with the monohybrid cross, it's going to be pure breds. Now, it's really easy to show dominant and dominant, recessive and recessive, but in reality, the purebred does not have to be big R, big R, big Y, big Y. It could be big R, little y, little y. But it makes it a lot easier to show it this way when you're learning. So P generation, big R, big Y, big Y. The homozygous recessive is going to be little r, little y, little y. Why are the parents like this? Because they're purebred. The F1 generation is when you cross this parent and this parent. You will get the F1 generation. How do you get the F2 generation? You will cross two of these to get the F2 generation. So just like in the monohybrid, when you cross the parents, you are going to get 100% big R, big Y, little y. They're going to be heterozygous across the board. That is what an F1 generation is. It's heterozygous across the board. So instead of drawing this out for you, they just went straight to the F2 generation, creating the F2, which is kind of, I wish that they didn't do that, but hopefully that's not confusing for you guys. So in order to get an F2 generation, we are getting one F1, and we're going to cross it with the other F1. And you might say, well, aren't they brothers and sisters? And I would say, yes, you are absolutely correct. But when we're dealing with plants or we're dealing with other animals, that is not as big a deal as if we're talking about humans. So, in order to draw out the different alleles at the top, each one of these tops has to have an R and a Y. And then each over here has to have an R and a Y, because that's how we're filling in the cells. And so this individual is, both of these individuals are, again, big R, big Y, little y. That is their genotype. And so with those 4 letters, what are all the different combinations you can make? Well, you're seeing it right here. You cannot make any other combinations with those letters of that individual. And so that is why they are written at the top, because those are all the combinations that you can create. Same with here. It's the same letters. And so we do the same thing here with this dihybrid cross. We're going to fill in the cells by bringing the letters over. And so, you know, big R, big Y, and big R, big Y, you're going to create double big R, double big Y. And you fill in all of these. And so I guess we're looking at the phenotype here, and I don't know if you remember, big R equals round and big Y equals yellow. So all of these cells, even though they all have different even though they all have different genotypes, all of them will have the phenotype round and yellow. When we look here, the phenotypes are round and green. When we look here, it is oval and yellow. And then this last one, which doesn't have a circle, is going to be oval and green. And so the phenotypic ratio is 9 to 3 to 3 to 1. I would say you definitely should remember for your practical and for tests that 3 to 1 ratio from the mono hybrid. That the 3 to 1 phenotypic ratio with the with the monohybrid. The 1 to 2 to 1 is also very important, and that's the genotypic ratio. When it comes to this dihybrid ratio, it's a little more difficult. I wouldn't ask, but you never know. But if you were to be given this without the colors circled, you should be able to figure it out, to circle it, to calculate it yourself, to come up with this phenotypic ratio. But the big one here is, you know, this particular circle. If I were to say, you know, what is the phenotype of this individual? And you would say that it is round and green. And then if I said, well, how many other round and green ones are there? Well, you could figure that out by looking at all of the cells. Human genetics. Some human traits are controlled by a single gene. I had brought this up before. Some are not. Some are controlled by multiple genes. When it's controlled by a single gene, some of these traits exhibit a dominant inheritance pattern. Dominance inheritance is when a trait is passed from son to daughter, son or daughter, by one parent. So, you know, if the father or the mother has this dominant allele and they mate with somebody to create offspring, that offspring is going to have the dominant allele expressed, because it doesn't matter if they're going to, whether that child is going to be homozygous dominant or heterozygous, it's going to express that dominant allele. Dominant traits are expressed or seen in heterozygous individuals, since the dominant allele masks the recessive. Um, some of the traits are recessive, meaning that they are masked. And so a recessive inheritance is the recessive inheritance is when a trait is passed to the son or daughter of both parents. So if a child is expressing the phenotype of a recessive allele, then that means they have to be homozygous recessive. They have to have both alleles will be small a. And that means those small a's came from both of their parents. The recessive traits are expressed only in individuals that are homozygous recessive for that trait. Now, the reason that we're bringing this up is we're going to get into pedigrees. Pedigrees are different tables that we see down here. They're drawings of family lines, looking at particular traits of that family line and how it shows up. And just like Mendel did all these mathematics, he looked at those ratios of when I cross this with this, I'm going to get this, and it's going to happen 75% of the time. That means, and he's doing all this math. Well, when it comes to human beings, we can't do that as well, because you're not just randomly mating them with whoever you want. These matings are happening in real time in their own ways. And so pedigrees are kind of, you're taking the reality of what's happening and kind of writing it down and looking for patterns that are going on in there. So pedigree is an analysis that is used to track the inheritance patterns in families. And how do we do this? Well, we have to have, they have to be set up in a way that, every time you draw one of these, the people know what you're talking about. There are standards to these pedigrees. So that means the symbols and everything have to be the same across the board. Symbols represent people, and those are squares, which are going to equal males. Circles are going to equal females. I do not know of a symbol that's the combination of the two. I am sure that there is something out there, but I guess they're just trying to be more simplistic with this and just show the males and females. If you ask, what's the combination, there could be many different combinations here of this male-female situation. But I digress. The lines, they are going to represent relationships. Horizontal lines are going to mean the matings of the two. Now, in a lot of what you're going to read, it's going to say the marriage of two. Now, we all know that you don't have to be married to an individual to have children, but some of these pedigrees are old-fashioned, they're old-timey, and so a lot of the times they will mean that marriage, but really it just means that there is a mating situation that happens. So the horizontal line is mating, but I will accept marriage if you give me that as an answer. Vertical lines means offspring. So this vertical line is showing that these are the offsprings. These three are the offspring of these two parents. And then, so this particular offspring of these two parents then mated with another and had these two children, both daughters. And then this daughter mated with this male and had 4 children, 3 boys and a girl. So what are the filled in and the empty symbols? Well, basically the filled in symbols represent the affected individuals. affected basically means that that is the phenotype that this pedigree is showing. So it could be red hair. And so this red haired female mated and had 3 children with not red hair. One of her children mated and had 2 children with not red hair. Then that child mated and had 2 children with red hair. And so that could be what it's showing. It could be showing all types of different things. But I'll give you a hint here. This is a recessive trait because it is not seen in every single row, and that is needed for a dominant trait. So this is a recessive trait being seen. An example of a dominant inheritance pedigree. Affected individuals must be seen at least affected individuals must have at least one affected parent. Phenotype usually appears every generation. Usually. And so, this filled in, this pedigree could be the dominant trait of brown eyes. And so, these 2 parents, we've got a blue eye and a brown eye. They have children. Even though it's dominant, we have we only have one of the 3 children with brown eyes. But if that were the case, what else could we learn here? Well, the fact that if this is a dominant pedigree, so that means this individual, this mother is having children that are not all affected. What do we know about her? We know that she cannot be dominant. She cannot be homozygous dominant, because if she was, all of her children would be affected. We know that she has to be heterozygous. And so that is why we have children that are being born that are not affected. But the dominant pedigree is showing that in every generation, the trait is being seen. In a recessive inheritance pedigree, unaffected parents can have an affected offspring if the parent is heterozygous for that trait. And so what that's saying is these 2 parents are not showing the affected trait. This particular trait we know is recessive, but if we didn't, we can easily learn that because these 2 are not affected by whatever trait we're looking at. They have children where one of them is affected. So that means if this were dominant, then if this were a dominant trait and they're not being affected. So that would mean that they have to both be little a, little a, little a, little a, because the dominant is going to be expressed. So I'm talking about how I'm kind of confusing myself. If this were a dominant pedigree, not a recessive pedigree, then the fact that they're not affected, the only way that that could be is if they were little a, little a, little a, little a. If they were both little a, they could not have an affected child. So we know from the bat that this cannot be a dominant pedigree. So if it were a recessive pedigree, that means, these individuals could be big A, big A, big A, big A, or if they were both big A, well then all of their children across the board would be big A, big A's. And so they're not big A's, but they could be heterozygous, big A, little a, big A, little a. And so that is the only way that these 2 can have a little a child. And so here we see that as a recessive pedigree because we're skipping a generation.